Hi everyone. Now we will be talking about our first topic, evolution. Evolution is something that a lot of people associate with Charles Darwin. And Charles Darwin was one of the main researchers looking into studying evolution. And so I wanted to take you through this picture here, which, by the way, is not a scientifically accurate picture at all. It's just used to illustrate a couple of ideas. So Charles Darwin, when he studied evolution, which again, if you remember back, the idea behind evolution or the definition behind evolution is that organisms change and adapt to their environments. And that's all evolution is. Evolution is the ability to change and hence adapt to your environment and be able to survive and pass on those genes to your offspring or your children. And so Charles Darwin, when he studied in the Galapagos Islands off the coast of South America, he noted several features in different types of organisms, and he came up with early ideas about evolution and natural selection. And natural selection is the theory by which evolution occurs. And so if you look at these two pictures, there's a picture on the right and a picture on the left, and it's used to illustrate an example of giraffes. And so this could illustrate how giraffes evolved over time. One theory is, or I should say, one hypothesis is that over here on the right, over evolutionary time, there were some giraffes with long necks, some giraffes with very short necks, and some giraffes kind of in between in terms of height. And the question that is posed in this picture is, how did giraffes get such long necks? And so this is an evolutionary question. So if you look at this picture, it's telling you some giraffes had really long necks early on in evolutionary time. Some had really short necks and some were in between. And what had happened is trees that grew with the food source required for these giraffes was quite up high. And so only the ones that had the long necks were able to reach the food source, and hence the other types of giraffes died out. There's a different hypothesis over here on the left-hand side. And this one says that initially there were giraffes that were shorter than their food source. And as the food source and as trees grew higher and higher, giraffes would have to stretch higher and higher. And based on that environmental pressure to stretch, their giraffes over time got longer and longer. These physiological changes were passed on to their offspring over time, over several years. And so there are these two ideas, and when you think about Charles Darwin, you want to think about which idea did he have. And so when you look at Charles Darwin, he actually leaned more towards this idea over on the right-hand side. Some giraffes had the long necks, and again, they were the ones that were best able to fit into their environment, so they survived and passed on the genes, and all the offspring got the long necks. There's another evolutionary researcher you don't hear a lot about, and he's named Lamarck. And I was taught about Lamarck in evolutionary class when I was an undergraduate. And his theories were often laughed at because it suggested that you could change your physiology by adapting to the environment. And that wasn't necessarily exactly the same as Darwin's ideas. The fact that you could stretch your neck out so much and pass those adaptations on to your children or your offspring was not well received at the time I was going through school. However, now we know more information about a field of study called epigenetics, EPI genetics. And epigenetics is essentially changes that happen above the genome. So they're not mutations in the DNA. They are mutations or changes that are happening on top of the DNA. So it can change how the DNA interacts. And what's amazing now, many researchers over the last 10 to 15 years have really delved into this field of epigenetics because it helps explain the causative nature of many different aspects of life, in particular disease and changes to physiology, like you see here. So, for example, epigenetic factors have been linked with cardiovascular diseases, cancers, etc., especially because in DNA, you need an exact mutation and you have to pinpoint that exact mutation in order to link it to a disease or a trait. Whereas in epigenetics, it's more so what's happening to you environmentally. How does that actually change the fluidity of your DNA? That's not passed on through a mutation or a direct change to the sequence, it's passed on through how the DNA interacts. And this is studied scientifically. It's a huge area of research, and we can talk more about it when we reach the DNA unit. So let's start evolution off by looking at some truths or lies. So individuals do not evolve, populations evolve. Is that a truth or a lie? So this one is a truth. Individuals don't evolve. You don't evolve on your own. It's actually a population as a whole that's evolving. So when we talk about evolution and the ability of a population to survive and adapt to its environment, that's what we speak of, the population in itself. Here's another truth or lie. Humans evolved from chimpanzees. And you'll see the answer later on in my slides. But this one is a lie. So humans did not evolve from chimpanzees. We are really closely related to chimpanzees. The latest numbers show us around 98% genetic similarity, which means that 98% of those 25,000 genes are shared amongst humans and chimpanzees, which is pretty amazing because humans to each other, we are already around 99.9% similar in terms of our DNA. And so chimpanzees are really close. Mice, I studied mice in my graduate degree, and mice are really close as well. They're around 95 to 96% similar to humans. So we didn't evolve from chimpanzees. We are genetically similar, but we actually came from a common ancestor. So that's where that similarity lies. But our lines diverged. So here's a common ancestor. Chimpanzees went one way, humans went another way. So we did not evolve directly from chimpanzees. Here's another statement. Evolution rarely follows a straight line from species to species. Instead, it is more like a tree with many branches. Some branches lead to new branches while others become dead ends. And that is true. So you can see that actually in the next slide. If you recall, this is the phylogenetic tree of life. And you can see that there are many branch points. This is not by any means a complete phylogenetic tree of life. It's very small, and it's just showing you some of the main organisms in each of these three domains of life, which again are bacteria, archaea, and eukarya. Animals or humans fall into this group here. So those are the three domains of life, again. So when we think about evolution, again, it is one of the theories in biology, the theory of evolution. And again, all it means is that organisms can adapt to their environment and then those adaptations can be passed on. So what are some pieces of evidence for evolution? In your book, you have four main pieces of evidence, and they are biogeography, fossils, and anatomy and embryology, and molecular biology. So biogeography is the idea that in different areas of the world, there are different organisms there. And so they have evolved on separate locations in the world. Fossils, so those are stones or rocks, different formations where you can see imprints, and those are known as fossils of different organisms over time. And you can date fossils using a method called carbon dating, which allows you to gauge how many millions or how many thousands of years ago a certain organism adapted to its environment. Anatomy and embryology is sort of shown here in this picture. So anatomy and embryology, and specifically comparative anatomy and embryology, tells you that if you look at a human's arm, if you look at a dog's leg, if you look at a bird's wing, and if you look at a whale's fin, they all do something a little bit different. So they function a little bit differently, but they're all really quite similar. And the fact that we have similar anatomy tells you that we must have evolved from a common ancestor, as well as embryology. So if you look at an embryo of a human, and if you compare it to the embryo of another organism, another mammal, or another animal, then they look quite similar. And so because of that similar nature, that tells you that there is common ancestry. You don't even have to just look at the differences. When you look at the genes that are involved in forming these structures, so genes are our underlying blueprint of DNA, and genes code for proteins, and proteins make up everything in our bodies. And so these limbs, for example, are made up by different proteins that are activated in embryogenesis, which is when we're embryos in the womb. And so the same can be said for other organisms. It's actually the same genes in a human's arm that are activated at the same time a dog's legs are, when the dog is developing in utero, and same for bird and whale. Um, so when scientists map out what were those genes involved in limb development, and if you look across these 4 different organisms, they're all pretty much the same genes. And that points to molecular biology as the last piece of evidence for evolution. So it's true that over several years, there have been studies published that show phylogenetic trees, and those phylogenetic trees are often grouped by looking at different traits. So back to the anatomy and embryology, looking at different traits. Do these 2 organisms both have hair? If so, they must be more closely related than another organism that does not have hair. Do these 2 organisms have a vertebral column? If so, they must be more closely related than one that does not. And so all of these phylogenetic trees have been presented in scientific literature over hundreds of years. But now that we have molecular biology, which is DNA evidence, now that we can actually go back and sample DNA, and for current organisms, we can do this as well, we can take a sample of the DNA and we can actually tweak those phylogenetic trees that were created a long time ago and see if the DNA does support how those hypotheses were laid out. And if it doesn't, if we find that other organisms were more closely related, that's where we can shift the evolutionary tree a little bit. So evolution in itself as a theory does not point to these different evolutionary trees. Those are more so hypotheses posed by various researchers around the world studying fossils, biogeography, and comparative anatomy. But being able to actually add the DNA evidence into there to solidify that, yes, for example, humans and chimpanzees, they are very closely related. Being able to do that actually adds a lot of validity to the study of evolution. And so that's what's known as the modern synthesis in evolution. It's the ability to take phylogenetic trees and back them up with DNA evidence. There are some misconceptions about evolution. So some people suggest it is just a theory, but remember going back to our discussion in the introductory biology unit that actually a theory is much more solid and there's evidence for it over hundreds of years and it has not been proven false yet, or it has not had evidence against it yet. Individuals evolve is not true. Again, it's populations that evolve, and organisms evolve on purpose. That's another misconception. So organisms do not behaviorally evolve on purpose. They will evolve based on their environment. So taking a look at climate change, for example, those organisms that are able to adapt to a quick changing climate, they will be able to evolve faster and they'll be able to survive and pass on their genes rather than those organisms who are unable to adapt to a fast changing climate. So like I mentioned, Charles Darwin is one of the main researchers when we study evolutionary biology, and he coined the term natural selection to suggest a few things. And he suggested that every organism within a population has its own individual fitness. And that fitness is essentially the ability to A, reproduce, and B, survive and pass on the genes that they have. So within a population, because remember it's populations that evolve, within a populations, individuals can still have their own reproductive fitness. And again, that's based on mating and survival. So Charles Darwin looked at a group of different finches in the Galapagos Islands when he was first studying, and he noticed that their beak shapes would change when the location of these finches was moved. So what had happened is there became a barrier within this group of finches that he was studying. And so when you take members of a group into a totally new habitat and you change the food source, what he noticed was their beak shapes had to change to accommodate the new food source. And so you can see these changes happening in these organisms. And that helped him to solidify some of his ideas on natural selection, the idea that you can adapt to your environment and be able to pass on those genes. He drew these early phylogenetic trees that you see over here on the left-hand side, and if you compare that to earlier, our 3 domains of life tree, it looks fairly similar in terms of how Darwin thought speciation occurred, which is formation of new species. And so if you look at a modern day elephant's phylogenetic tree, you can see his ideas were not very far off. If you take a look at the Galapagos Islands now, these are some pictures directly from the Galapagos Islands. You can see that the Galapagos Islands house many species that are endemic to there and that you cannot find anywhere else in the world, which again is evidence for evolution. So you find these giant tortoises, and you can actually book an adventure cruise to go and look at these different organisms in their natural habitats, and it's a protected area. So you find different species, different animals, and you find these really interesting birds called boobies, and specifically they're called blue-footed boobies. And this is a baby booby, and what's really interesting is that they've retained some of their own instinct because they have not been exposed to other organisms outside of this area. And even though they have unlimited resources in the Galapagos, because again, it's a preserved habitat, so even though they have unlimited resources, what happens is that when you look at these boobies, so here's a mom blue-footed booby, when they have eggs, so let me skip ahead, they have eggs, so mom and dad, blue-footed boobies, have 2 eggs here. What happens when the baby boobies are born is that a little while later, they will actually fight to the death. And so they have this instinct to fight in order for one to survive. So over evolutionary time, mom and dad can only support one blue-footed booby, even though right now they would be able to support 2. But however, they still have that biological instinct to fight to the death because previously there would only be able to be one supported. Going back, you probably saw some pictures of these boobies. These are red-footed boobies, called for their red feet versus the other ones that are blue. And there's some more pictures of different organisms that exist in the Galapagos and some pretty interesting adaptations. So moving on a little bit, I wanted to discuss the reproductive barriers to speciation. So I mentioned the word speciation, and that's how an organism becomes a brand new species. So we don't just get new species popping up in different parts of the world all the time. In fact, there's a lot of barriers to actually developing a new species. So when we look at these barriers, we group them into prezygotic barriers and postzygotic barriers. So what prezygotic means is, firstly, the word zygotic or zygote means early embryo, and prezygotic means before that. So these are barriers that exist before an embryo is actually formed. Postzygotic barriers on the next slide would be after an embryo is actually formed. What are the barriers to speciation that exist? So here I've listed some of the prezygotic barriers to speciation. So temporal isolation means that two organisms, what happens with them, two different organisms mate at different times of the year. So maybe there's two species of frogs here, these two, and they could potentially mate together and create a new species of frog, but they are temporally isolated, meaning, let's say this frog only wants to mate in the springtime and this frog only wants to mate in the fall. Because of that, they'll never produce a new offspring or a new species. So you could potentially get them together, but if they don't want to mate at the same time, then you won't get a new species. Behavioral isolation is essentially how it's defined by behavior, is that the female in a species, she prefers the behavior of a male of her own species versus a different species. This is actually something that I looked at when I was in undergrad, and I looked at fruit flies. So fruit fly mating behavior is a huge topic in scientific literature, and essentially I was looking at a female and a male of the same species mating together and how fast that mating occurred versus a female of that species with a totally different species of fruit fly. So there are different species of fruit fly, but the question is, can you even create a brand new species by mating them together? So that's something to look at with that type of prezygotic mechanism. So behavioral isolation is specifically female choosiness in terms of the behavior of the male. And again, usually females prefer members of their own species because of that. Mechanical isolation is that the reproductive organs of the two species don't physically fit together. Gametic isolation is the fact that the two organisms actually are able to mate together, but the early embryo or the zygote actually does not form because something happens with the gametes, and the gametes are egg and sperm. So they may come together, but they don't form a zygote. So it's like a miscarriage. So, for example, miscarriages in humans are often defined as those embryos that cannot form because of incompatibility with chromosome number. And so gametic isolation is that similar idea that the two gametes, the egg and the sperm, cannot physically come together. Habitat isolation is just as the name suggests. Maybe here, for example, in these two species of grasshopper, this one prefers a different habitat than this one, and so just based on location, they'll never come together to create a new species. When you look at postzygotic mechanisms or barriers to speciation, there's two: hybrid inviability and hybrid sterility. So hybrid inviability and hybrid sterility, both of those postzygotic mechanisms happen after the individual is born. So let's say you are able to take two members of different species together and mate them, and you do have an offspring. That offspring is either inviable, so they're weak, they're feeble, and they die early on, or that offspring is sterile. So that is in the case of a mule, for example. When you take a horse and a donkey, usually they're sterile. And so that tells you that you can't really create a new species because then you don't have any organism to breed with them. So something else to consider when looking at speciation is, how is the speciation occurring? So there are methods called allopatric speciation and sympatric speciation. So when you take a look at this left-hand side picture, you can see an owl population. And so when you separate the owl population here in yellow and separate them from other organism, another group within them, and you move them down to a totally separate region, then they're exposed to different environmental cues. And so there's a physical barrier that doesn't allow those organisms to interact together. So, like a land barrier, or oftentimes a mountain is usually a physical barrier, or a stream, or some sort of water. So when you have a physical barrier in between those two populations, they can't interbreed back to each other. And so they may become a new species over time, like Darwin's finches. And so when that speciation happens based on a physical barrier, that's called allopatric speciation. When you have an organism within water, let's say, so you have an aquatic organism like this fish, when there is a genetic mutation that occurs while in the midst of the parent population where there's no physical barrier, let's say this one genetically mutates to have different shape of lip, then you can potentially get a new species. That can happen, and it's very rare. It's called sympatric speciation. It's where a new species arises right in the midst of its parent population or the original population. So this new species is actually still just in the realm or in the same geographical location as the original population. There's no barrier, but it just became a new species due to a spontaneous genetic mutation. That can happen as well. The rates of speciation can be different as well. So if you look at this top panel of this picture, gradual speciation versus punctuated equilibrium means something a little bit different. So if you look at time here, and if you look at this term gradual speciation, just by the meaning of the word gradual, what that means is that this initial founder species gradually changes over time into two new species. So that's what's known as gradual speciation. You get a few little changes that happen over time that lead to new species. What punctuated speciation or punctuated equilibrium means, let's say you have that same starting species, the founder species, but then you have a drastic change right away, and that change is maintained over time, and that leads to the new species. So punctuated means early change and it's maintained further, whereas gradual speciation occurs with very small changes over a long period of time. There's a lot of numbers on this slide, and this slide points to populations and population genetics. So when we think about evolution, there's a field within evolution that's called microevolution, and microevolution looks at genetics. It looks at something called allele frequencies, and alleles are just different versions of genes. And so you could have different versions of a particular gene based on what you inherited from your parents. So for example, eye color is one of those. So there is a gene for eye color, and then you either inherited the blue eye color allele or the brown eye color allele, for example. And so we can apply alleles and how they're inherited to populations and look at microevolution. So there's a formula called the Hardy-Weinberg principle that looks at different possibilities of alleles and how they change in populations. So here we have an example of garden peas. So these are the three alleles that we see in garden peas, and garden peas are either yellow colored or they're green colored. And so yellow color is what's known as dominant in the population. That's the color that we see to a higher degree in that population of garden pea than we do of the green peas. And so we have more of the yellow, so we call it dominant, and the green, there's less of it, so we call it recessive. And so if you use the letter Y to illustrate the alleles, you would use the letter capital Y to illustrate the dominant yellow color and the lowercase y to demonstrate the green color. And so those individuals or those garden peas that are yellow would have two capital Y's because you get two alleles, one that came from mom and one that came from dad in sexual reproduction. And so remember, you have to have two alleles for a gene, for any gene that you have. And so you're going to see two capital Y's for the yellow piece. You could alternatively see a capital Y with a lowercase y, where the dominant Y, it masks the effects of the recessive Y. So even though we inherited a little y here for the green color is actually masked because you have one capital Y. The one capital Y is enough to give the yellow color. And then you have two little y's here on the green pea. And so what scientists do in microevolution is they look at the numbers. They look at what percentage or what amount out of one of the population, so this is a fraction of one or a percentage of one, what amount is actually the two capital Y's, what is the one capital, one lowercase, and what is the two lowercase. The two capital Y's, we call them homozygous dominant. The one that has the mix, but the dominant color is called heterozygous. And the green one over here is called homozygous recessive for the fact that you have the recessive color. So you can see just based on these numbers, there's a lot less of the recessive ones, homozygous recessive, than the other two. And so scientists can study how populations are changing based on what the numbers are of these different types of alleles in the population. So there's nothing in here that you necessarily need to memorize in terms of numbers or process, but this is just an equation, and again, it's called the Hardy-Weinberg principle that's used in microevolution to study how these alleles are changing in a population. And that gives you insight as to how they're passed on to their offspring and what traits are passed on to their offspring. So you can do this type of analysis, and this is called a Punnett square. If you've learned a little bit about inheritance and genetics, you may have seen Punnett squares before, but essentially this is how you would go about the process. And eventually you have this formula called p squared plus 2pq plus q squared equals 1, and you may see this formula come up in when we discuss genetics later on, but this is the evolutionary formula in microevolution that's used to assess allele frequencies. And then based on doing this type of analysis, you get your numbers that you saw earlier on, and you can see how many of the offspring in a population might have the dominant color versus the recessive color. And so this is how you can analyze something called genetic drift. So again, this is the same idea, but over generations. So this is a generation of, or a population of bunnies. And you see that there's a lot of bunnies that are homozygous dominant and then heterozygous for the brown color of fur color. So the gene is fur color. Brown is the big B allele. And the little b allele over here is the white fur color for these bunnies. And so there's only two in this population that are white colored. After rabbits reproduce within their population, you can see that over time we are weeding out this recessive color. So essentially, as you get more rabbits interbreeding with each other, the chances that the brown ones interbreed and produce more offspring are higher. And you can actually map that with these numbers. So you can see these numbers that relate to the formula P and Q from Hardy-Weinberg. And so scientists that are looking at microevolution can actually apply these numbers, and based on the numbers they can see how the alleles are changing in a population. You can also just look at the population and see how it's changing, but but scientists who study microevolution want to know what the alleles, what's happening with the genetics behind this population. And so again, over time, you have more rabbits reproducing. Let's say these two that are highlighted here reproduce more, and over time, you've weeded out this white fur color from the population. So remember I mentioned to you that in evolution, it's true that populations evolve over time, which we can see here that we've evolved based on fur color. However, each of the rabbits have their own individual fitness. So these two happen to mate a lot more, and hence the next population of rabbits, the next generation was representative of that. Now, this isn't the only way how populations can change over time. There's something called the bottleneck effect. And so if you apply this to a population, it's in the form of a natural disaster that can occur. So let's say you have those rabbits and there's a forest fire. So we have different colors of marbles in this bottle, and this bottle has a small opening. And so in a forest fire, maybe just a few members of the original population survive. But are they going to be representative of the past population? Not necessarily. So let's say the past population has these colored marbles, these light orange colors, darker orange colors, and these green colors. So let's say in the original population, there were a lot of the light orange, lots of the green, and only a few of the dark orange population. So if you flip that bottle and you allow 6 marbles to come out, for example, 6 or 7 marbles, your question is, will that be representative of the original population? And again, not necessarily, because in a natural disaster, the forest fire, doesn't care what alleles they're weeding out of the population. So it could be just that the light colored orange and the green marbles survive, and we don't have any members that are this darker orange color. And so that's something called the bottleneck effect. It's essentially genetic drift where alleles are changing in populations over time, but it's quickened because of a natural disaster. And so this is your new surviving population that doesn't have the alleles for this particular color. Of individuals. So that can happen in populations as well. Gene flow is another way by which organisms can migrate and introduce new alleles into a population. So here it's very simple. We have a group of brown beetles. Here we have green ones. And this one here has decided to migrate and allow its genes to flow into this population. And now we've introduced new genes into the population. So that's called gene flow, and it happens due to migration. The other way, again, is to summarize with mutations that can occur. So sometimes you could get spontaneous mutations in populations. Lethal mutations can exist. So lethal mutations is where you have a mutation in the DNA, but it unfortunately leads to the death of that individual. Beneficial mutations are spontaneous mutations that happen, but they are allowing that individual to have some sort of benefit in the population over the others, whether that be benefit of survival or reproduction, whichever one can occur. But this is very rare. It's not common for beneficial mutations to just spontaneously arise in populations. Neutral mutations are those that are most common, and neutral by definition of the word are mutations that have no effect on survival or reproduction of the organism. And finally, I wanted to mention adaptive evolution. So adaptive evolution is how a population can change based off of its original population. And so this is the same idea as the previous ones that I've shown you so far, like gene flow and genetic drift are the mechanisms behind them. But then what does adaptive evolution actually look like? So if you take this panel up here, for example, this blue line is what's called a bell curve. and a bell curve occurs for any population of organisms. So let's use the rabbit example. So if we take the examples of rabbits, which are actually down here, let's use these ones here and follow them through all three panels. So these three rabbits exist in these three fur colors. So we have this gray color over here in the original population. Most rabbits are this white fur color in this particular population, and then very few are mixed white with black. And so we have very few of those. So we have very few darker colored rabbits, lots of white colored rabbits, and then very few that are mixed with both. So this population of rabbits happens to follow this blue bell curve. And again, this bell curve is based on numbers. So you would plot numbers of the organisms here on the y-axis. And so when something happens in the population, like one of the mechanisms that was discussed that allows gene flow or allele flow, the population can change. So when we select for only the individuals that have the white fur color, let's say, we get this new curve. And this new curve takes into account that you don't have any of these individuals on the extremes. So none of the individuals that are the grayish fur color or the white and black fur color. So we've weeded those out from the population. We just have a population of white furred rabbits. And maybe that's an advantageous fur color in that particular population, because if you think of, for example, the Arctic habitat, it is advantageous to have white fur color because you can blend into the snow. And so maybe in that population, we get this effect of stabilizing selection, where we stabilize and only select for a particular group of members of that population based on fur color. Directional selection, if we look at, again, that blue original bell curve, what directional selection means is that only one of the extremes is favored. In this picture, the one over here on the right-hand side is favored. So we generate a new red bell curve where we have more members of this extreme group of species. So for example, if we took this white and black rabbit, this was on the extreme end here. In this new population over time, the red bell curve, we have more of those white and black white and black rabbits, just because the environmental pressure has selected for it that way. And that's called directional selection. You could have alternatively had a red bell curve over to the left-hand side here, and you'd be favoring these grayish rabbits in that population. So if you need to blend into the environment, maybe it makes more sense to be a gray colored rabbit in that particular environment. And so you would have a new bell curve that mimics or that shows that there's an increase in the individual here that's on the extremes. Diversifying selection is where you have environmental pressure to actually favor both extremes and not the middle. So here you can see this new red curve shows you that we have a high increase in the black and white rabbits, and we have a high increase in the grayish rabbits, but a sharp decline in the white rabbits. And so that tells you something is happening in the environment that pressures these two fur colors of rabbits, but not the intermediary. Maybe their predators can better see them, and so we see a sharp decline in the amount of white rabbits in that population. So I wanted to also mention the sexual selection hypothesis, the idea that this is called the good genes hypothesis. We've talked before about mating a little bit, where we talked about prezygotic barriers to speciation. And so you kind of already know about mating. And so here's a few pictures of individuals where there's a female and a male of the species. And again, female, male. And what you notice is that the males are often very nicely colored. And that forms an evolutionary purpose, because females are often the choosy ones in the species where they select the males. And remember, evolution tells you that males want to be able to survive and pass on their genes to the next generation. And same with females. So females are going to be choosy. They want their offspring to have the best genes. So they're going to choose the best male. And so the best male comes in the form of competition. So if they're stronger than other males, and in the form of behavior. So if they're able to present themselves and do a proper courtship ritual or dance for the female. And again, how they look. So you can see these brightly colored feathers here in these different organisms. They're very attractive to the females. And so that leads to the good genes hypothesis, that there must be some sort of genes underlying or telling the female through a physiological appearance that this male has good genes. And of course, the female wants good genes for her own offspring. So that is known as the good genes hypothesis. And again, that points to more of the individual competition and the individual fitness of organisms within a population. So that Drosophila mating behavior video, I actually have a link to it here. And so I can show you that that sort of sexual selection exists. And this video doesn't have sound, but it does show you a male and a female mating together. So there is a courtship song. The male is here, the female is here. And the female, again, is the choosy one. She will choose the male whether she wants to be mated with the male or not. And so in this case, there is successful mating, and you can study this in a lab like I did, and you can study this and see if the female chooses different males of different species. Now, the next example is a male, but he's with a previously mated female. So she's already been mated by another male. And you can see that in this example, she's rejecting the male and running away, and she will not allow mating to happen. So that shows you again that females are the choosy members of the species, and they decide based on several cues as to which males she will allow for mating with. So you can see that the male is following, but the mating is not successful. Now this leads me to a discussion on something called the sweaty t-shirt gene. So I'm not going to play this video for you, but you are welcome to watch it. So I was really interested in this type of mating behavior, and something that I was taught was about a gene called the sweaty t-shirt gene. And that is actually the name of the gene. It has been called the sweaty t-shirt gene. And so there are scientists trying to figure out why human females are attracted to certain males. And so what scientists did is they had males wear around t-shirts until they became sweaty, and then had females smell these various different types of t-shirts. And then the females would choose which ones were the best smelling to them. And what was really interesting from that study, and the video shows you, is that the females would often choose the t-shirt smell that was most different from her. And when they tried to figure out what gene that was involved in, it is a gene that's important and responsible for the immune development within humans. So the female actually chose the male sweaty t-shirt from the male that had different immune system genes than she did. Why is that advantageous? If you then mate the male and the female, their offspring would have a variety or a hybrid of different immune system genes that would allow that offspring to function better in its environment and have better resistance to pathogens like bacteria and viruses. So that sweaty t-shirt gene really does have some sort of not only physiological response, but also it has something to do with our allele combinations. So that's important to look at in terms of putting together the microevolution component along with the more macro evolution component. So moving on a little bit to phylogeny, this is the last portion of the evolution unit. So again, going back to these evolutionary trees, scientists have developed these evolutionary trees over several hundreds of years. And they can be, a lot of them that I'm seeing now are more of the circular trees, but some are still this kind of linear format. And they do follow a specific format if you look at different studies. And you can see these different evolutionary trees. And usually what you're going to see is some sort of line leading to a common ancestor, and then as you move upwards or sideways, then you get to later in geological time. And then you see different branches depending on what domain of life it is or what kingdom of life it is, and then how these individuals segregate. And evolutionary trees or phylogenetic trees used to be constructed where you would ask questions like, do these two organisms, if I'm going to group them together, do they both have a vertebral column? And if the answer is no, then one of them diverged, and the other one we can ask more questions of and see if they're related to other organisms. So questions like, do they have hinged jaws or legs or eggs with amniotic fluid, or do they have hair? And if no, then that means they're less related. If yes, they both do have hair, then they are more related. And so this was an early method by which you can construct evolutionary trees. And that was based on whether the different physiology and anatomy of these organisms, whether they matched other organisms. Again, now with modern day techniques looking at DNA evidence, we can better match individuals as to whether or not they share ancestry. Taxonomy is the way by which we call organisms what they're called. So here we have a specific type of dog, and its specific name is Canis lupus familiaris. And so when we look at taxonomy, that's the specific scientific name for this particular breed of dog. And how we look at that organization is again from top to bottom, starting with the domain and going all the way to the bottom to the species and subspecies. So how a dog would be grouped is within the domain Eukarya. Eukarya contains both contains animals, and so the kingdom would be Animalia. Then we would break it down into a phylum, a class, and the class is mammals, the order, which is carnivores, family, genus, species, and then subspecies. There's nothing in particular here you need to memorize, but just be aware that taxonomy is the way by which we group organisms. So again, we can look at similar structures across different organisms and analyze their relatedness. This is called homology. So for example, a bat wing is used for the same purpose as a bird wing, and they look fairly similar. And so we can then group these organisms closer on an evolutionary tree. We can also look at analogy. So are they used for similar processes? So are they used for flying, for example? And yes, in this case they are. So these would be related more closely to each other. But what's interesting is these insects here, they have a different type of structure to that wing shape. And so because of that, they would be grouped further away from these two individuals. And if you look at penguins, for example, they have wings as well, but they are not used as flight instruments, so they would not be closely related to these organisms. So I'm putting it quite simplistically, but there's really a lot of research that goes into trying to put together phylogenetic trees in a particular way. So animals, and there's nothing in particular here in terms of eras and periods you need to memorize, but animals really didn't show up until much later in geological time. And so when you think about time in itself, several millions and millions of years ago, we know there was the existence of prokaryotes, and prokaryotes are very simple cells. like the cells that make up bacteria. And so prokaryotes were existing much earlier in evolutionary time than what's called eukaryotes. Eukaryotes are much more complex types of cells that make up animals, for example. So it wasn't until the Cambrian explosion where we saw this huge increase in organisms. And so the Cambrian explosion you see somewhere around here, uh, that's really when animals came into the world. And in terms of geological time, that's really not that long ago from now versus pre-Archean time, which was much, 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 as you can see in the number in terms of millions of years ago, much earlier than that. So the Cambrian explosion again was somewhere between 500 and 540 millions of years ago. And so over these different periods, the complexity of animals has certainly increased. And we can see evolution happening over time based on evolutionary evidence, which I pointed to before, like fossils and comparative anatomy. So again, that Cambrian explosion period was when the diversity, the biodiversity, increased over time. And so this huge increase of different animals was essentially born into the world. We have lots and lots of fossils based on carbon dating that have been dated to the Cambrian explosion. So that's where scientists believe the first animals really appeared. There have been mass extinctions over time. So if you look at millions of years before present time here on the x-axis, and then extinction occurrence on the y, you can see that in certain periods over time, there have been mass extinctions. And so essentially, as you look over time, there have not only been these explosions of animal phyla, for example, but also mass extinctions. And you can see a cycling to these events. So over geological time, we know that there will be cycling. And if you take a look at primates, you can see, for example, there's 5 here, one of which is us humans. And you can see that in terms of our anatomy and comparison across these primates, you can see that we're really quite similar, but there are also a lot of differences. And so based on comparative anatomy, we can group these individuals into an evolutionary tree like this. So here we have baboon, gibbon, orangutan, gorilla, and the chimpanzee. And here we have lines that lead to common ancestry amongst all of these. And way down here you have humans and then a bunch of different early humans like Homo erectus and Homo neanderthalans. And so if you take a look at humans and if you take a look at chimpanzees, you can see that as you go back over time, you can see that there is some common ancestry. And all of this is based on different skulls and remains that scientists have found over time. So the Lucy skeleton was one of those different skeletons of Homo erectus, for example. And based on comparative anatomy and comparative skulls, for example, we can group organisms in such a way to suggest that we are more closely related to these early humans than we are to chimpanzees that exist now. However, we do have common ancestry several thousands of years ago with chimpanzees. So we are more closely related to chimpanzees, and if we look at humans, we are further away from the baboon, for example. So if you go back even more in evolutionary time, we did share common ancestry, but that is much further back. That's why we are more closely related to chimpanzees than other primates. There's nothing specific about this picture you need to memorize, just some of the details that I mentioned about relation to chimpanzees versus other types of primates. So as a discussion and as a class activity that you can work on following this evolution unit and chapter, either in groups on the discussion thread or on your own, you can try and answer some of these questions on evolution based on our discussion. And these are quite application-based questions. So firstly, what was the Cambrian explosion that I mentioned? And why do you think it was important for speciation and diversity? Based on our discussion, how does diversity affect adaptation? Give an example of an organism that has adapted to its environment by diversifying, and there's lots of examples. What is homology? So I briefly mentioned what the word homology was, and why is it important for constructing evolutionary trees? What is sickle cell disease? So this is something I did not discuss, but if you look it up, sickle cell disease is one of those diseases that's recessive, and I talked about alleles and what recessive versus dominant means. And it's really interesting because when I mentioned alleles, there's the homozygous dominant individual and the homozygous recessive individual. And the homozygous recessive one, when a disease is recessive, would have the disease. The heterozygous individual would mask the effects of the disease, but it's really interesting if you take a look into the origin of heterozygous individuals for sickle cell disease and what link there is with malaria. So see if you can use a scholarly search engine. Again, the library is a good resource, or Google has a scholarly search engine, and it's called Google Scholar. And so if you search in Google Scholar even, you will, and if you search for the sickle cell gene and malaria, you might find something really interesting about those heterozygous individuals. Why might it be good to carry a diseased allele with a chance of passing it on to your children or offspring? Why would it be good in areas that have high malaria infection? Think about why that happens evolutionarily. Explain how antibiotic resistance in E. coli, which is a type of bacteria, exemplifies evolution by natural selection. And antibiotic resistance happens over time. It can happen when you're, for example, prescribed antibiotics for something that is a viral infection, or when you're not taking the proper dose of your antibiotics, you're not finishing it, or you're taking someone else's antibiotics. This is a huge problem nowadays where we see the bacteria have actually developed resistance to the antibiotic, which means it's not working anymore. How does that exemplify evolution for the bacteria? Why is that a good or bad thing for the bacteria? And then this one is purely subjective. So which traits do you think are being selected for in a human population? And why do you think they're being selected for? So that's something you can discuss with the discussion thread or think about on your own from this lecture. So thank you again. If you have any questions, please feel free to email me.